Hello students, I hope everyone is well following the health rules. Welcome to my zoology class. This is Akmal Hussain, lecturer biology, Square High School and College. Now I am going to take a zoology class of standard 12 chapter 10, immunity of human body. Today's topic is second line immune system. What will we learn from this class? We will be able to know about role of macrophage in destroying bacteria, role of neutrophils in destroying bacteria, steps of phagocytosis. Second line of immunity. Last class we have discussed about first line of immunity of human body. If first line fail to protect our body from different pathogen, this pathogen entered into our body, then second line of immunity starts its activity. If a pathogen exceed the first level of immunity, the second line stays prepared to neutralize them in order. This line consists of white blood corpuscles, tissue, blood vessels, phagocytes and lymphocytes. Different types of cell of second line of immunity: mast cell, natural killer cell, monocyte, macrophage, neutrophil, etc. Role of macrophage and neutrophils to destroy bacteria. Bacteria can infect a human through several portals of entry in our body, like nostril, mouth, eye, ear, etc. Macrophages and neutrophils helps to destroy the pathogens and keep the body free from infection. Number 1, macrophage. Macrophage comes from Greek word Markos, means large, and phagein means eat, that means big eater. Actually, macrophage can take or kill the large size of bacteria or pathogen. That's why it is called macrophage. Macrophages are a type of white blood cell of immune system that engulf and digest cellular debris, foreign substances, microbes, cancer cells, and anything else that does not have the type of proteins specific of healthy body cells in its surface in a process called phagocytosis. Phagocytosis is a process through which the WBC, especially the macrophage and neutrophil, can kill the microorganism into our body. Characteristics of macrophage. Number 1, 4% of total WBC. Number 2, size 60 to 80 micrometer. Number 3, it stays lung, liver, kidney, connective tissue, brain and lymphatic duct. Number 4, it takes 100 bacteria at a time. Secretes cytokine. What is cytokine? Cytokine are biochemical substance which is secreted by certain cells of the immune system and have an effect on other cells. Functions of macrophage. It kills pathogen, this is the main function, as scavengers. They rid the body of worm out cells and other debris. Along with dendritic cell, They are foremost among the cells that present antigens, a crucial role in initiating an immune response. Dendritic cell. What is dendritic cell? Dendritic cell is another type of cell which take part in second line of immune response, and most of the time they stay the beneath of our skin. Number 2, neutrophil. Neutrophil are type of white blood cell that is characterized histologically by its ability to be stained by neutral dyes and functionally by its role in mediating immune response against infectious microorganisms. The microorganism which are responsible for different type of infection is called infectious microorganism. Characteristics of neutrophils. Its diameter is 12 to 15 micrometer. 60 to 70% of total white blood corpuscles. Shows pseudopodial movement. Produces from bone marrow. It takes 3 to 20 bacteria, then it die. 100 billion are produced per day in our body, this type of cells. 50% stay in tissues and left 50% in blood vessels. Functions of neutrophil. Neutrophils are a kind of white blood cell filled with enzyme. They help phagocytic cells kill and digest microorganisms that they have engulfed. Neutrophils are the body's main immune response to assault on the body by bacteria and fungi. Phagocytosis process. The ingestion and killing process against bacteria or other microorganisms by phagocytes is called phagocytosis. Phagocytes, the cells or white blood corpuscles which take part in killing the microorganism by engulfing, is called phagocytes. Phagocytes here macrophage, neutrophil, etc. Phagocytosis process. There is a picture of phagocytosis process. This is a phagocyte and this one is bacterium. At first, phagocytes contact with the bacterium, then make pseudopodia, and finally both pseudopodia or two pseudopodia attached to one another. Thus the bacterium entered into the body. This is called ingestion. Then this bacterium entered into the body and starts some activities. By the help of digestive enzyme of lysosome, digest the bacterium or kill the bacterium or break down into small pieces. Then the phagocyte excrete the wastage of the bacterium. This is the phagocytosis process. The total phagocytosis process we may divide it into 7. Dear students, today's class is descriptive, so you have to read a little bit more. Step 1, name activation. This step of phagocytosis occurs when the cell comes in close proximity to given object. For instance, when phagocytes are near such cell as bacteria, this this activates the phagocytes and stimulates binding. This first step also involved with chemotaxis. Step 2, name ingestion. The cell starts expanding as it surrounds the molecule. This process also involves formation of a vacuole or vesicle around the molecule as it is completely ingested. There is a picture of ingestion. Step 3, formation of phagosome. Actin filaments send pseudopodia out to engulf the microbe and place it in an endocytic vesicle called phagosome. Follow the picture. This is bacterium entered into the neutrophil or macrophage cell. When bacterium entered into the body, this condition is called phagosome. Step 4, formation of phagolysome. Lysosomes are vesicles that contains hydrolytic enzyme that breaks down molecules. A phagosome fused with a lysosome is called the phagolysosome. Follow the picture. This is phagosome and this is lysosome. When phagosome and lysosome fused one another or attached one another, this condition or this situation is called phagolysosome. Step 5, intercellular killing and digestion of bacteria. Bacteria secrete two types of chemicals by combination with lysosome and phagosomes for destruction and digestion. Proteolytic enzymes are secreted from its lysosome to digest bacteria or other foreign protein molecules. This enzyme is sufficient to digest most of the bacteria. Step 6, residual body containing indigestible materials. Phagosome is enclosed by a membrane, so bacteria can't enter the cytoplasm. It may stage of ingestion. However, if the appropriate nutrition element is absorbed at the phagosome's cytoplasm. Final step, discharge of waste materials. Cellular waste such as broken down molecules that the cell can't reuse is discharged from the cell by the process of exocytosis. There is a picture of phagocytosis. Let's watch a video of phagocytosis. Stay at home, stay safe. Thank you.